So would the protein be translated from the messenger RNA? Yes, it would, and most of you realize that, because the information's still there for the ribosome to dock on. The protein's going to start being translated. It's just going to lack that ER signal sequence. And without anything, without an ER signal sequence, SRP wouldn't bind, that whole thing wouldn't happen. It won't be translated into ER, but be shorter because it lacks the ER signal sequence. And Dr. DeLuzan brought up this possibility, which really could happen. So why might this be a correct answer? Why might that mutant protein be ubiquitinated and degraded in the proteasome? Go ahead.
This is a sizable hydrophobic patch.
It's lacking that hydrophobic patch, so we've removed the ER signal sequence. But, go ahead.
It's using the chaperones inside the ER lumen?
Right, so it's using the HSP70-like chaperones in the ER lumen, and those, it's not going to see those. Instead, it's going to see the cytosolic HSP70s. So presumably, maybe this is what you were going to say too, it's not folding properly, and what happened to misfolded proteins? They are ubiquitinated and sent to the proteasome. It's also lacking, we're going to go through this later, but most of the proteins that are going to be in the ER are glycosylated, and it would not be glycosylated and protected, it wouldn't be folded properly, all of that. So odds are actually 4 is probably the correct answer, but that's a subtle answer. Yeah.
Is pH inside the ER any different than from the cytosol?
Absolutely is. So it is more similar. So as we'll see later, that as we go through the secretory pathway, the pH gets more and more acidic. So the pH of the ER lumen is more neutral. It's more similar to the cytosol, but it's still a little bit different. The other thing that's drastically different about the endoplasmic reticulum is it's full of calcium. And the cytosol is, of course, quite low in calcium. So this different environment could also affect the folding of the protein. Good point. Other questions? Okay, so we've gone through a protein that's going to be secreted. So let me use this little thing to review what we just said. So we have a protein. So I'm a ribosome right here. Okay. Here's the big subunit and the small subunit coming on. So here's the protein. So I'm translating this protein. So this protein is being translated. Here's the positive charges that emerge. Here's the ER signal sequence that has emerged from the ribosome. And now SRP comes, binds that, stops translation. All of this stops until we come to the SRP receptor on the ER membrane, right here, the ER membrane. SRP receptor binds, takes it over to the translocon. So then this ER signal sequence transfers to the inside of the translocon. This takes off, and translation continues. So translation's continuing, and the protease will take off the ER signal sequence. Translation's continuing, and that ends up folded, there it is on the floor, folded in the ER lumen, right there. Okay, so that's how all secreted proteins begin life, with this ER signal sequence. And really, it's this, with our question?
Yeah, I have a small question. So I was just wondering, so does the SRP bring it to the translocon, or does the translocon go to the SRP?
Excellent question. So how do these things move?
Right.
And remember that all membranes have high lateral mobility. So it's a matter of just lateral diffusion. So it's neither one bringing it to the other or the other bringing it. They sort of bump into each other. Now it turns out that it's not, the ER membrane doesn't have these proteins evenly dispersed throughout them. Instead there are islands where these proteins are restricted, but they're laterally moving in there. So the probability of them bumping into each other is higher. But it's still just random motion of lateral movement. Good question. Other questions? Okay, so it's these same ingredients then that we use to explain how a protein So we've explained how a protein that's going to be free in the extracellular matrix, this could be a protein like growth factor, collagen, fibronectin, anything that's going to be on the outside of the cell is going to begin life in the ER. And it turns out these same ingredients with some addition are how proteins are made that are going to end up not secreted to the outside, but end up on the plasma membrane, sort of like the protein that we discussed in our first example right here. And in order to understand that, we first think of the conundrum of this protein. So we have a protein here. This is a protein on red blood cells that I think we have seen before. This is glycophorin A, an integral membrane protein. And somehow the cell is faced with the problem of putting this synthesizing a protein so it has its transmembrane domain jammed into a membrane, ending up on the plasma membrane with some kind of another independently folding portion on the exterior surface and another independently folding portion in the cytosol. And it's these same ingredients that cells use to synthesize a protein into the membrane. This protein like this that's going to be on the plasma membrane is actually going to be synthesized initially into the ER membrane. And then once it's in that membrane, it will never leave membranes. It's going to go in vesicles through the Golgi and in vesicles up to the plasma membrane, always having this integral membrane portion, integral membrane domain embedded in a bilayer. So in order to do that, it's these same ingredients plus a couple more that explain how the 20 or so amino acids that we know reside in the transmembrane domain end up there. So let's look at that. And let's also think about all the different kinds of proteins that are on the plasma membrane. Where the cell has to face complications of a protein that spans the membrane with its amino terminus in the cytosol, its C terminus in the ER lumen, its C terminus in the cytosol can go in the other opposite orientation, and it can span the membrane more than once. It could span the membrane 7 times like this one, or 9 times, or 12 times, or 3 times, any number of times. So all of those variations exist. So how does the cell synthesize complicated proteins like this? And how does it determine whether the N terminus or the C terminus is going to be on one side or the other? And that's what we're going to look at now in the last 15 minutes. In order to do that, we need to add 2 more, well, it's really 1 more ingredient. And this is the nomenclature, the terminology that Albert's used, but it's a little bit, it's helpful, but it's confusing in some ways. So in order to do that, we need to add a stop or a start sequence. These will end up being dictating the transmembrane domains. I think my sound isn't on anymore.
It's on.
Okay, thank you. Transmembrane domains. And these are on the protein being synthesized, on the integral membrane protein. And what's confusing about this is it sounds like they're different sequences, but they're actually the same thing. It depends on how they function. So let's go through this and you'll see what I mean. So the start and transfer sequence are going to end up being the transmembrane domains. So you already know what that sequence is. It is 20 or so non-charged, generally hydrophobic amino acids. You know what a transmembrane domain is. So you know what the start and stop sequences are. And they're going to end up being the transmembrane domain. And when these sequences are translated, they can either initiate, depending on where they are, or stop the transfer of the emerging polypeptide through the translocon. So they'll stop or start, depending on where they are, the protein as it's being synthesized, and they'll end up being the transmembrane domain. So let's look at how that works. And we're going to start with a protein that, like this, that passes the membrane a single time and has its amino terminus in the ER lumen and its C terminus on the cytosol of the ER, of the endoplasmic reticulum. So in this diagram, as in many of the diagrams are simplified, so the SRP is left out of here, the SRP receptor is left out of here, the ribosome is left out of here, just to simplify the diagram. But you all should know that all of those are there. So what we see here is just the messengers left out, so it just shows the protein being synthesized. So just as we saw before, in this particular case, this is going to end up being a protein that is that has its amino terminus on the. So if we look at the ER membrane right here, it's going to span the membrane